Good morning, everyone. I'm Jia Tong from Beijing National Day High School, and it is my pleasure to have the opportunity to attend the Sigma Psi Student Summit. Today, I would like to present the poster of my research assessing surfactant absorption during disinfection process. First of all, I would like to introduce the background and purpose of my study. During the COVID-19 pandemic, we need frequent disinfection to prevent the spreading of the coronavirus. Surfactant is an important active component in many disinfectants we use. It is an amphoteric molecule, which means that it has a hydrophobic tail and a hydrophilic head. During a disinfection process, the surfactant first attaches its hydrophilic head to the surface of bacteria or virus, which carries electrical charge, and then it embeds its hydrophobic tail into the lipid membrane or protein capsid and kills the microbe. We can see that the surfactant-microbe interaction has a direct impact on the disinfection effects. My research aims to assess the absorption efficiency of different surfactants and find the dominant factor that affects surfactant absorption. In order to complete these experiments in a home lab, I applied simplified and feasible models as substitutes for real microbes. The diagrams below shows the conversion from the real disinfection mechanism to the home lab models. I use silicon dioxide powder to simulate the lipid membrane of bacteria, which carries negative charge, and calcium carbonate powder to simulate the protein capsid of viruses, which carries positive charge. Here are the experimental methods of the research. At the beginning of the experiment, I prepared the solid powders and samples of surfactant solution. The surfactants I use are benzalkonium chloride, which is an cationic surfactant, sodium lutecyl sulfate, which is an ionic surfactant, and primary alcohol ethoxylate, which is a non-ionic surfactant. For each surfactant, I prepared 6 samples with varied concentrations. Then I measured the refractive index of each sample and found the linear correlation between the refractive index and concentration of each surfactant. Then I began to add solid powder to the surfactant solutions. I stand the samples for 48 hours so that the solid powder can precipitate and thus will not affect the following measurement. And at last, I measure the refractive index of the remaining solution in each sample after the 48 hours. And I find their final, or we can say the equilibrium concentration from the previous correlation I established. From those data collected, I calculated the amount of absorption, which is the number of milligrams of the surfactant absorbed per gram of solid powder. Here are the results of the study. There are 2 types of absorption models that can apply to this research: the multilayer adsorption and monolayer adsorption. In the multilayer adsorption model, the solid powder attracts multiple layers of surfactant molecules, thus the amount of absorption increases as the concentration increases. In the monolayer adsorption model, because the solid powder attracts only one layer of surfactant molecule, the amount of absorption will not increase once saturated. I converted the absorption data into these graphs that show the relationship between the amount of absorption with the equilibrium concentration for each surfactant. For the cationic surfactant absorption with the silica powder, we can see that the amount of absorption increases as the equilibrium concentration increases, which corresponds to the multilayer adsorption model. And for the absorption of the ionic surfactant with the calcite powder, we can see that as the equilibrium concentration increases, the amount of absorption has no significant alteration, which correspond to the monolayer adsorption model. For the non-ionic surfactant absorption with the silica powder, we can see that as the equilibrium concentration increases, the amount of absorption increases, which also correspond to multi-layer adsorption model. Here are the conclusions of the study. So in this study, I was able to establish a valid home lab to simulate the surfactant microbe interaction during disinfection process and quantify the absorption by the correlation graph of concentration and refractive index. We can conclude that the cationic surfactant bacteria and non-ionic surfactant bacteria pairs undergo multilayer adsorption, while anionic surfactant virus pair undergoes monolayer adsorption. We can also conclude that the electrostatic attraction is a dominant factor to the surfactant microbe adsorption. In order to achieve high efficiency disinfection, we highly recommend cationic surfactant containing disinfectant for bacteria and anionic surfactant containing disinfectant for virus. For example, during the COVID-19 pandemic, the anionic surfactant containing disinfectants are recommended to use and promote to the society. Our future work can focus on the dominant factor of mixed surfactant adsorption. That is the end of my presentation. Thank you very much.